Hello and welcome. Today, we're diving into one of the most incredible stories in all of science. It's about how we, as humans, went from simply working with nature to actually learning how to rewrite its most fundamental code, the code of life itself. Let's get right into it. Okay, I know, it sounds a bit like the start of a really strange joke, right? But the answer connects some of the most ancient things we do with the absolute cutting edge of science. The common thread here is our amazing ability to understand and, well, work with biology on a whole new level.
So first up, let's talk about biotechnology. Now, this isn't about creating something totally new out of thin air, not at all. It's about being clever and using the biological machinery that nature has already spent millions of years perfecting.
I want you to think of this as the main theme for our whole discussion. On one side, we have harnessing nature's processes, which is all about collaborating with living things just as they are. And on the other side, we've got rewriting nature's code. And folks, this is a revolutionary jump, where we become the architects of biology itself. All right, here's the official definition, and for those of you studying for your O-levels, this is a really key term to lock in. Biotechnology at its core is about putting living things, especially the super tiny ones, to work for us, to get them to make stuff we need or carry out jobs we want done. So why microorganisms? Why these tiny little guys? Well, because they are pretty much the perfect employees. Think about it. They're small, they reproduce at an unbelievable speed, and they can perform these really complex chemical reactions that are super difficult, if not impossible, for us to do in a lab. You know, from the yeast that makes our bread fluffy to the bacteria that turn milk into yogurt, we've been relying on this unseen workforce for thousands of years. And here's where it gets really cool. This slide breaks it down beautifully. We're looking at enzymes, the protein workhorses made by these microbes. Got cloudy apple juice? Well, the problem is a substance called pectin. The solution? An enzyme called pectinase that clears it right up. Those stubborn stains on your clothes? The problem is proteins and fats. The solution? Proteases and lipases in your washing powder. Can't digest milk? No problem. The enzyme lactase can break down that lactose for you. It's problem solving at a molecular level. But okay, how do you go from a tiny sample of microbes in a petri dish to producing enough for the entire world? You use one of these, a fermenter. You can think of it as a super high-tech, perfectly controlled home for microbes. Everything, and I mean everything, from the food, the pH, the temperature, even the oxygen levels, is managed precisely to get them to work as hard as possible for us. And this brings us to Act 2. We're about to take a huge leap. We're moving from just using nature to actively rewriting it. This is really where science fiction starts to become science fact. You see, for centuries, we had to just find the right organism for whatever job we needed done. But then, starting in the 1970s, scientists figured out something incredible. They learned how to take a gene, a specific piece of DNA code, from one organism and stick it into a completely different one. This is genetic modification. We didn't have to search for the perfect microbe anymore. We could actually build it. So how on earth is this actually done? Well, this slide breaks down the whole process perfectly. Step 1, you identify the exact gene you want, let's say the human gene for insulin. Step 2, you use these amazing things called restriction enzymes, which act like molecular scissors to cut that gene out of the human DNA. Step 3, you take a small circle of DNA from a bacterium called a plasmid and you cut it open with the same scissors. Step 4, you use what's basically molecular glue, an enzyme called DNA ligase, to paste the human gene into that bacterial plasmid. Step 5, you pop this new combined plasmid back into a host bacterium. And finally, Step 6, you put that engineered bacterium into a fermenter, where it starts multiplying like crazy, becoming a tiny factory that pumps out human protein for us. It's just amazing. And that special piece of combined DNA, it has a name, a recombinant plasmid. The word recombinant just means it's a new combination of DNA from two different places. You can think of this plasmid as the delivery truck. It's the vehicle that carries the new set of instructions into the host cell, telling it what to build. Okay, so we know the how, but now let's get to the wow. What has this unbelievable power to rewrite the code of life actually allowed us to do out here in the real world? Well, the impact on medicine has been nothing short of a miracle. I mean, before genetic modification, insulin for people with diabetes had to be taken from the pancreases of pigs and cows. It was expensive, it wasn't a perfect match, and it could cause allergic reactions. Today, genetically modified bacteria produce a pure, limitless, and absolutely identical supply of human insulin. It saved millions of lives. And it's not just medicine. Agriculture has been totally transformed. Take these herbicide-resistant soya plants. It means farmers can spray their entire field with weed killer, and while all the weeds die, the soya plant, which has this special resistance gene, is perfectly fine. Or think about Bt maize. It has a gene from a bacterium that makes a toxin that's deadly to insect pests, but harmless to us. This means farmers don't need to spray nearly as many chemical pesticides.
And this example right here, this shows the incredible humanitarian side of this technology. Vitamin A deficiency is a massive problem in many parts of the world. It can cause blindness and even death, especially in children. So to fight this, scientists engineered what they call golden rice. They inserted genes that make beta-carotene. That's the stuff our bodies use to make vitamin A. It's a powerful, targeted solution to a huge public health crisis.
But, as you can imagine, with great power comes great debate. This technology, especially when it comes to our food, has sparked some really intense controversy and a lot of concern all around the world. So, let's take a balanced look at the arguments. It really is a question of trade-offs. On the pro side, GM crops can absolutely lead to higher yields. They can reduce the need for farmers to use harsh chemical pesticides and herbicides, and they can lower costs. But on the con side, there are some very real concerns. The high cost of the patented seeds, the risk of creating herbicide-resistant superweeds by accident, and the potential for unintended harm to other insects, like butterflies or bees. So, despite all this ongoing debate, the data tells a very clear story. Just look at this graph. It shows the percentage of GM crops grown in the USA since 1996. You can see this incredibly steep, rapid adoption by farmers for both herbicide-resistant soya and pest-resistant maize. So what's the big takeaway here? It's that while the public debate continues, the economic and practical benefits for the people actually growing our food have been so strong that it's driven a nearly complete shift in modern agriculture. So let's just bring this all together for a moment. We've seen two massive revolutions. First, there was biotechnology, where we learned to harness nature's tiny engines, using microbes to do things like bake bread and clean our clothes. And then came genetic modification, where we started rewriting the blueprint itself, creating designer medicines and superpowered crops with abilities they never had before. And this leaves us with a final and pretty deep question. For all of human history, our limits were set by nature. Now, for the first time, we have the tools to set our own. The ethical questions, the societal challenges, they're every bit as complex as the science itself. And that's a conversation that all of us really need to be a part of. If you found this explainer helpful and learned something new, please give it a like and be sure to subscribe to Spotwork Learning. We've got a lot more deep dives into the world of science coming your way very soon. Thanks so much for joining us.